Good morning, house. I hope you had a beautiful Valentine's Day celebration yesterday. Last week, I sent a note on our Module 2, which is factors affecting the growth of microorganisms in food. But before we discuss the factors affecting the growth of microorganisms, let's discuss microbial growth. Growth can be defined, microbial growth can be defined as an autocatalytic process. There's no growth will occur without the presence of at least one viable cell. That's viable, a living cell. And the rate at which the growth will increase with the amount of biome, it will depend on the amount of biome present. That means, for example, if you have 4 cells in a microbial culture, and you have another microbial culture that has 1 cell. So the rate at which the viable microbiome will increase in the 4 cell will be different from how it will increase in 1 cell. Bacteria reproduce by asexual process called binary fission. That's fission, splitting into 2. And in this series of events, the chromosome duplicates and the cytoplasm splits into 2 equal halves, each with 1 chromosome. Then the mesosome may be involved as it grows out of the lateral surface of the cell membrane. If you have seen, of course, you should know the structure of the microbial, of a bacteria, the simplified version of a bacterial cell. There's an invagination of the mesosome, and that's where the division starts from. It extends out while the chromosome now splits into 2 when the cell wall completely forms the division, or that sign of breakage. Now, different bacteria have their generation time, what is called doubling time. For some, it can take as long as 33 hours. Some can be 30 minutes. Some can be 20 minutes. And a good example of the one that is in 20 minutes is Escherichia coli. And that's why most the genetic engineering, in order to make more copies of a particular product, for example, insulin, E. coli is used. The chromosomes are cloned, and it is incorporated, what is called genetically modified. And as it replicates or divides, the more copies of the E. coli is being made, and more synthesis of the insulin. So that takes us to a microbial growth curve. What happens in a microbial growth curve? Now, the growth curve will only happen in a closed system, what is called a batch culture. There are basically 2 types of system, or 2 types of culture: the batch culture and the continuous culture. In the batch culture, the microorganism will go through the 4 phases of growth, which is the lag phase, the log phase, the decline phase, the stationary phase, and the decline phase. That will only happen in a batch culture. And what does that mean? It means that nutrients are only supplied once to the culture, and that's a batch. But in a continuous culture, you keep supplying nutrients to that culture while you harvest your metabolites. So it depends on your target products. For some companies, their target products will be produced at the logarithmic phase, which is the log phase, while some other companies will harvest their product at the stationary phase. So you will not allow the cell to enter into the dead phase, because you keep adding. Once they get to the log phase or the stationary phase where you need your target products, you add nutrients so that they don't die off. So what are the characteristics of the log phase? The log phase is the initial phase where the cells get acclimatized into the new environment. You are bringing them from a different environment. You want to introduce them into a culture that is new. So they need to familiarize themselves. Sorry about that. So as I was saying, in the lag phase, that is the first or the initial stage where the microorganisms need to acclimatize themselves to the environment. And in that phase, there's no growth, no apparent growth, but there's synthesis of enzymes that are required, for exploitation as well as repairs of any lesions resulting from earlier injury. For example, freezing, drying, heating. Probably you brought out your inoculum from a freeze-dried um, a freeze-dried microbial isolates. So they will need to acclimatize, to the new environment. Then they they might have been subjected to drying or heat. They will still need to synthesize enzymes to repair their injury. So in that phase, there's no multiplication. No multiplication. So multiplication is 0, death is 0. The place is new. So then the duration of the lag phase now will also depend on the amount of the inoculum that was added to the medium, as well as the environmental condition of the medium. So that will make the lag phase either short or long. Then the microbial cells will now enter into the next phase, which is the logarithmic phase, the log phase. And here, the microbial cells enters an active stage, and that active stage is known as the log phase, and is often shortened, as log phase. That's logarithmic or log. Now, the cell rapidly increase in mass and they reproduce by fission. This is where they begin to divide. The population is at its biochemical optimum, and research experts are often performed at this time. For example, if in a pharmaceutical company where maybe penicillin is being produced and inoculums are added and the cell begin to grow or begin to multiply. Now, what the cells release as metabolites, or what we call waste product, is that products, that the company needs. Either microbial products could be antibiotics, could be gas, it could be acid, it could be there are 7: acid, gases, antibiotics, forgotten them now. Anyway, depending on your company. So if yours is antibiotics, at that stage of log where the metabolites are released, all you need to do is to harvest, and you add a fresh batch of nutrients to the medium. Now, the microbial cell from the exponential phase now ends in the next phase, now enters the next phase, which is the stationary phase. Now, the stationary phase is as a result of key nutrients being depleted or inhibitory metabolites accumulated, and the cells now move into stationary phase. Now, when metabolites are released, because it's a culture system, metabolites are released Now, the metabolites that are released may now begin to inhibit the growth of the microorganism in that culture. They are becoming toxic to it, to the cells, if they are not harvested. And that can now lead to the next phase, which is the stationary phase. Now, in the stationary phase, nutrients may become scarce, waste products may have accumulated, or the medium may have become dehydrated. It's just because it has now become, it's no longer the normal rich environment where the microorganisms are familiar to. So the death and multiplication is not the same thing. As they are multiplying, they are dying. As they are multiplying, they are dying. So that's what we mean by stationary phase. So there's no increase in the number of cells again. And this now takes the medium into the next phase, which is the death phase or the decline phase. And this is overwhelmed, when the population, due to the accumulation of toxic waste and the depletion of nutrients, the population now becomes overwhelmed and the bacterial cells begin to die. But some of them, if they are capsulated microorganisms, they now form capsules which protect the cell, from the environment and makes them to survive longer. Then for organisms that have flagella, which is an organ of locomotion, they may decide to move away from the medium to locate areas where you have nutrients. They are trying to survive, so they move to another area where they can actually find food to eat. Then at this stage now, the bacteria can also produce spores, which is the restive stage of bacteria. And these spores can actually survive hard or harsh environmental conditions. So if the bacteria through history of the population soon comes to an end with death of all cells, so that's the end stage of the phases, the death phases. Then most viable cell in that phase will die. But in continuous culture, don't forget that you keep adding nutrients. So there's no way that culture will actually enter into the decline phase. It is either you harvest your metabolites or your products at the log phase or at the stationary phase. This is actually what happens in the bacterial growth curve. And there's a curve. I'm going to send the picture of the bacterial growth curve to you right now. So now, what are the factors that affect microbial growth in food? The first factor we're going to be discussing is grouped under chemical factors. And the first factor under that is nutrient content. Microorganisms use food as a source of nutrients as well as energy in order to grow and function normally. So the microorganisms also of importance in food require water, source of energy, nitrogen, vitamins, and related growth factors, as well as minerals. Molds have the lowest requirements. They have the lowest requirement for of these nutrients, followed by yeast, then gram-negative bacteria, followed by gram-positive bacteria. So as sources of energy, foodborne microorganisms may utilize sugars, alcohol, amino acids, and few microorganisms can also utilize complex carbohydrates such as starches, cellulose, as their source of energy by first degrading them or breaking them down into simple sugars. In O-level biology, said disaccharide or polysaccharide is broken down into monosaccharide or disaccharide. So these compounds are actually broken down into simple sugar to make them easy to be utilized by this microorganism. So fats are also used by some of these microorganisms as sources of energy. But these compounds are attacked by a relatively small number of microbes in food. That's the gram-negative bacteria and molds. The gram-negative bacteria and molds are able to synthesize most or all of their requirements. So consequently, these two groups of organisms may be found growing on foods low in vitamins B. Fruit tends to be lower in vitamin B than meat, and this fact, along with the usual low pH and positive ER, which is redox potential in fruits, help to explain the usual spoilage of these products by molds rather than by bacteria. So most fruits and vegetables are prone to mold spoilage than bacterial spoilage. Why? Because of the low pH. That means most fruits are acidic, and except for the lactic acid producing bacteria, other bacteria will not be able to survive the bacteria that is produced in acid producing foods. And that's why you find molds as their major spoilage organisms. So that's for nutrients. Basically, nutrients in food serve as a good substrate for microorganisms because they are able to break down these nutrients and able to utilize them for their own purpose. So they will to make them grow, and that's where you find that they can spoil your food. So another factor that affects the growth of microorganisms in food is antimicrobial constituents. The stability of some foods against attack by microorganisms is due to the presence of certain naturally occurring substances that have been shown to have antimicrobial activity For example, most of our spices, most of our vegetables, they have antimicrobial properties that makes them it's natural in them, just like we have the innate immunity in us that makes it possible for us to fight against invading antigens or invading microorganisms. Now, plants also have this innate um, immunity in them as a result of the composition or the constituents that is present in them, and that makes them antimicrobial. An example is eugenol that is found in cloves. Cloves, you can Google it to actually see the picture of cloves. It is actually added to pepper soup as spices. It is added to most locally made drinks. Very good in when you add it to zobo drink. Tiny plants like flour, dried, dried flour that's what it's called, clove. Another um, component is found in garlic and it's called allicin. And that allicin in garlic makes the garlic antimicrobial. Cinnamic aldehyde and eugenol, they are also found in cinnamon. Then the milk of cow also contains several antimicrobial substances such as lactoferrin, conglutenin, lactoperoxidase, as well as casein. So these are some of the free fatty acids that occur in milk that makes them antimicrobial. So what these substances, what they do is that they protect that food product from invading microorganisms. But you find out that milk can also be subjected to spoilage. Now, a lot of factors will, because milk are supposed to be kept in the fridge, not freezer, fridge. Now, when you don't store your milk properly, of course, spoilage organisms will grow. You have tampered with the you have abused, the environmental conditions that could actually make. So what happens is, just like the fact that we have antibodies does not mean we should go and be drinking dirty water just because you feel that, okay, your immune system is intact, it will protect you from such things. You don't risk your life because you want to challenge your system. Another example is lysozyme that is found in egg, as well as milk also. Lysozyme is found in egg and in milk. Then there's also an enzyme that is called corn albumin, and this provides fresh eggs with fairly efficient antimicrobial system. Then in fruits, you have a compound called hydroxycinnamic acid derivatives. These are found in fruits, they are found in vegetables, they are found in tea, what you call your green tea, different spices, molasses, and other plant sources also show this antimicrobial as well as antifungal activity. A lot of vegetables that we use, ephedrine, bitter leaf, different types of vegetables, they have this natural, occurring antimicrobial substances that help them to evade or to fight against spoilage organisms. So natural constituents. We are still talking about factors that affect the growth of microorganisms in food. So when these factors are present, the antimicrobial constituent, of course, it will protect the food from being spoiled. Another factor under the chemical factor is pH. And it has been established that most microorganisms grow best at pH values between 7, that's the neutral, between 6.6 to 7.5, while fewer grow below 4.0. But bacteria tend to be more fastidious in their relationship to pH than molds and yeasts, with pathogenic bacteria being the most fastidious. And it is a common observation that fruits generally undergo mold and yeast spoilage. And this is due to the capacity of these organisms to grow at pH values of less than 3.5. I said they are highly acidic, so they can survive that acid environment, which is considerably below the minima for most food spoilage and food poisoning bacteria. So most meats and seafoods have the final or ultimate pH of above about 5.6 and above. So this makes this product susceptible to bacteria as well as mold and yeast spoilage. So most vegetables have pH values than fruits, and consequently vegetables are subjected to bacterial than fungal spoilage. So most foods are characterized by inherent activity. Others owe their activity or pH. Others owe their acidity or pH to the actions of certain microorganisms. So the latter type is referred to as biological activity and is displayed by products such as fermented milk, regardless of the source of the acidity, and the effect on keeping quality appears to be the same. We are going to explain biological activity in a short while. Now, some foods are able to resist changes in pH than others. Those that tend to resist changes in pH are said to be buffered. Meats are most highly buffered than vegetables. Contributing to the buffering capacity of meats are their various proteins. The proteins that naturally occur in makes them, when you say the person is buffered, it's like you have another shield, a protective shield, against spoilage organism. So that protein now acts as a buffer, as a shield for the meat against spoilage bacteria. So vegetables are generally low in proteins, and consequently, they lack the buffering capacity to resist changes in their pH by the growth of these microorganisms. So another factor is the moisture content. We are still talking about factors that affect the growth of microorganisms in food. So moisture content, under moisture has to do with the presence of water in the food. One of the oldest method of preserving food is drying or desiccation. And precisely how this method came to be used is not known. The preservation of foods by drying is a direct consequence of removal or binding of moisture, without which microorganisms do not grow. So water requirements of microorganisms is defined in terms of water activity in the environment. And this is defined as the ratio of the water vapor of pure water at the same temperature. And there's a formula for that. So in general, how does that affect us as a food microbiologist? Bacteria require higher values of water activity for growth than fungi. That means bacteria will grow very well in a moist environment. That's why you find them to be spoilage organisms of what we call perishable foods. Foods with high moisture content, such as fruits, vegetables. And this point is the gram-negative bacteria that have the highest requirements than the gram-positive. So in moist food, you find more of gram-negative bacteria as spoilage organism. So most spoilage bacteria do not grow below water activity of 0.19, while spoilage modes can grow as low as 0.80. Now, water activity also have a range or a chart, like a chart, and there are values for bacteria, fungi, that's molds and yeast. Just like you have pH scale, you have the pH value from 0 to 14. Water activity also has its own. So just as yeast and molds grow over a wide pH range than bacteria, the same is true for water activity. So certain relationships have been shown to exist between water activity, temperature, and nutrition. First, at any temperature, the ability of microorganisms to grow is reduced as the water activity is lowered. So once you lower the water activity of a food, the ability of microorganisms to actually grow in that food will reduce because they need moisture to actually survive. So the range of water activity over which growth occurs is greatest at the optimum temperature for growth. The optimum is the best temperature for growth, and that's at that temperature, water activity as well as the presence of nutrients will also make microorganisms to survive. So they are interwoven. Water activity, temperature, nutrient, they work hand in hand for microorganisms to actually survive in the food. The next point is oxidation reduction potential. Oxidation reduction potential of a substrate may be defined generally as the ease with which the substrate loses or gains electrons. This is chemistry. So when an electricity or a compound loses or gains electrons, the substance is said to be oxidized, while the substance that gains electrons becomes reduced. So oxidation may also be achieved by addition of oxygen. So a substance that readily gives up electron is a good oxidizing agent, and the one that readily takes up electron is a good oxidizing agent. So for anaerobes, that's microorganisms that grow in the presence of no oxygen. They require negative oxidation reduction EH value. So they are reduced when they are found in the food substance. So the redox potential of a food is determined by four characteristics. Number one, by four factors. The characteristic redox potential of the original food, the poisoning capacity, that's the resistance to change in potential of the food. That's what we mean by the poison capacity. It has the resistance to change in potential of the food. Now, the oxygen tension of the atmosphere about the food, as well as access that the atmosphere has to the food. So some aerobic bacteria actually grow better under slightly reduced conditions. And these organisms are often referred to as microaerophiles. Example of such, you have lactobacilli and streptococci. Now, some bacteria have the capacity to grow under either aerobic and anaerobic condition. Then you also have facultative anaerobes. Most molds and yeast encountered in food are aerobic, although a few tend to be facultative anaerobes. I'm going to give you a picture of this classification. When you have a tube and you have a nutrient broth and you introduce organism and inoculum into that nutrient broth. Now, if you want to know that these organisms are aerobes, by the time you incubate, you find them floating at the top. That means they want oxygen. They can only survive when there is oxygen. But if they are anaerobes, you find them at the bottom of the tube because they don't need oxygen. So they descend down to the bottom of the tube. So the microorganism affects the redox potential of their environment during storage, just as they do to pH. So aerobes can lower the EH of their environment, while anaerobes cannot. As aerobes grow, oxygen in the medium is being what? Depleted, resulting in the lowering of EH. So under redox potential, just remember that it has to do with the availability of oxygen in the environment. And when there are oxygen, you find that aerobes will thrive well because they need that to grow. And when you have the environment that is more of carbon dioxide, then you find the anaerobes to be the predominant factor that affects the uh, the growth of microorganisms in food under physical factor. We are done with chemical factors. Now, physical factors. The first factor there is the biological structures. Natural occurring of the natural covering of some foods provides an excellent protection against the entry and subsequent damage by food spoilage organism. For example, in humans, our skin is our natural covering and is the largest organ in the body. When the skin is intact, we are safe. On the skin, you have natural or normal flora that are acting, to protect us. But once there's a crack, once there's a break, once there's a cut on the skin, then the normal flora that is supposed to protect the body find its way into the deeper tissues and they become pathogenic. So that also occurs in food. The structured as the tester of seeds, structures as the tester of seed, the outer covering of fruits, the shell of nuts, the hide of animal, and the shell of egg. All these are the biological structures that are providing protection for that particular food. The outer shell and membranes of eggs, if intact, prevent the entry of nearly all microorganisms when stored under the proper condition of humidity and temperature. So fruits and vegetables with damaged covering undergo spoilage faster than those that do not. Example, your Esha tomato. When you go to the market and they tell you, oh cheap, it's just kò tí bàjẹ́, it's not yet spoiled, you can still manage it. They are cheaper. They have been broken. Sometimes you find molds already growing, or you see these white things flowing on the on the tomato. That's mold. They are prone to spoilage than tomato that is whole, that is not broken. The shell, the skin is still intact. Another example is the skin covering of fish and meat, such as beef and pork. And this skin actually prevents contamination and spoilage of these foods, partly because it tends to be dry. So it tends to dry out faster than freshly cut surfaces. Now, whole meat tends to uh, survive or tends to stay longer than meats that are cut into bits. When you cut them into bits, you have created an opening for microorganisms to enter, and it makes it easier for them for spoilage. So another factor under physical factor as well as extrinsic factor, external factor. The first one we discussed is the chemical factor, and it can also be regarded as the internal factors because it happens within the food. But this one is happening outside the food, and that's why it is extrinsic or external or physical. Is temperature of storage. Now, it is customary to place microorganisms into 3 groups based on their temperature requirement for growth. And these organisms are that grow well at temperature below 7 degrees and have their optimum between 20 degrees and 30 degrees are referred to as sacrotrophs. They are referred to as sacrotrophs. Now, those that grow well between 20 and 45 degrees with optima between 30 and 40 are referred to as mesophiles, while those that grow well at and above 45 degrees with optima between 55 to 65 are referred to as thermophiles. The sacrotrophs are found mostly in foods that belong to the genera. Sacrophiles found on foods belong to the genera Pseudomonas and Enterococcus. So when you store your food in the refrigerator and you still find that they go bad, the genus of that are responsible for this is Pseudomonas and the Enterococcus. This is the bacteria family Enterobacteriaceae. They belong to the family Enterobacteriaceae. Now, these organisms grow well at refrigerator temperatures and cause spoilage of meat, fish, poultry, eggs, and other foods normally held at this temperature. So some of these organisms can grow over a range of 0 to 30 or above 30 degrees. Example is the Enterococcus faecalis. Now, most thermophilic bacteria of importance in food belong to the genera Bacillus and Clostridium. Bacillus and Clostridium are good spore formers and they are thermophile. When you say thermo, that means they are heat lovers. They can survive high temperatures. Now, under this temperature, we also have some that are grouped as extreme thermophiles, extreme sacrophiles. The extreme sacrophiles are the ones you find in freezer, and the extreme thermophiles you find them. When I was introducing the course about habitats of microorganisms, we mentioned those extremes. Now, for Bacillus and Clostridium, their major the major place where you can find them is in canning industry. Even though those canned foods are subjected to pasteurization, but that heat is not enough to kill spores. So most molds are able to grow at refrigerator temperature. Notably, some of the strains of Aspergillus, Cladosporum, which are found growing on eggs, they are found growing on the sides of beef, and they are also found in fruits, are the ones that will actually spoil these your products even when you put them in fridge, the molds. You find them with like tiny patches, black patches on your fruits. Yeast also grow over the sacrotrophic and mesophilic temperature ranges, but generally not within the thermophilic range. So the quality of the food product must also be taken into consideration in selecting storage temperature. If you want to store your food, you must also put in mind the quality of the food products. The temperature of storage is the most important parameter that affects the spoilage of highly perishable foods. Highly perishable foods. I said foods are categorized into perishable, non-perishable, and semi-perishable foods. So most spoilage organisms, you find them in highly perishable food. Highly perishable food means they have high water activity. They are not resistant to spoilage. Example are fruits, vegetables, meat, fish, So they have a very short shelf life. But the semi-perishable have moderate shelf life. They are also moderately resistant to spoilage, and they have low water activity. But the non-perishable foods, they have very, very low water activity. They are resistant to spoilage, and they have long shelf life. And that's why your grains, your seeds, dry foods, basically, they are resistant to spoilage. So another factor, you are still considering the external or the physical environment where the food is stored, is the relative humidity. Now, the relative humidity of the storage environment, earlier we discussed the relative humidity within the food, the RH factor within the food. Now, the RH factor outside the food, that's the environment where the food is stored, is important from the standpoint of water activity within foods and the growth of the microorganisms at the surfaces. Now, when the water activity of food is set at 0.60, it is important that this food is stored under conditions of relative activity that do not allow the food to pick up moisture from the air and thereby increase its own surface water activity to a point where microbial growth can occur. So, for example, where do you store your grains? You store your grains in your store, dry place. If that's your stuff, if let's take a bag of rice, for example, you want to store your bag of rice, it's either you open up the bag and pour out the rice into a dry container, or your beans, for example, you pour it in a dry container, or if you place that bag of rice on the bare floor, cemented floor, after some time, you will find that your bag of rice will now be having something like, they will clump together, patches, green or black in color. That's because Yoruba will say the ground has breathed on your rice. Otimisi. Now, it's moisture that has been absorbed from the environment of storage, that is now causing that dry product to become moist and makes them prone to spoilage. That's what we mean by relative humidity of the environment. If the relative humidity of that environment is high, that your dry food will absorb the moisture and it will grow bad. It will go bad. So the relative humidity, there's a relationship between relative humidity and temperature that should be borne in mind in selecting proper storage environments for the storage of foods. Foods that undergo surface spoilage from molds, yeast, and certain bacteria should be stored under conditions of low relative humidity. So in selecting the proper environmental conditions of relative humidity, that's what you should put in mind. So consideration must also be given to both the possibility of surface growth and the desirable quality to be maintained in the foods in question. So by altering the gaseous atmosphere, it is possible to retard surface spoilage without reducing or without lowering relative humidity. Now, for example, if you want to store your beans, you can put them in the container, in a tight container, and put in the fridge. That's to prevent weevils anyway, to prevent weevil infestation. But if you now put them in the container and you leave the container open and you put the beans in the fridge but the container is open, after a while that your beans will grow bad because the fridge is moist. The relative humidity in that fridge is very high, and the moisture in the fridge will settle on your beans, and the beans does not need moisture, so spoilage organism will grow. Then you find that your beans is changing color, is having mold growth. You find these white patches on it. So another factor still under storage factor or physical factor or extrinsic factor is the presence and concentration of gases in the environment. So the storage of food in atmosphere containing used amount of carbon dioxide up to about 10% is referred to as controlled atmosphere or modified atmosphere of storage. Now, this modified atmosphere of storage is now used in major companies that deal with seafoods. So those seafoods are packed you see them like a plastic bag. But in that plastic bag, the modified atmosphere has been incorporated into the product so that it will not allow spoilage organisms to actually grow in those seafoods. It's a new area that is being worked on. I don't know if we've started doing, I don't know for any company that does that in Nigeria, although most of our things are imported anyway. So the concentration of carbon dioxide generally does not exceed 10%, and it is applied either from mechanical source or by the use of dry ice, which is called the solid CO2. That one is available in the market, dry ice. You can use them to preserve your sample. If you go for sample collection, maybe you go and collect your sample in Oyo State and you need to transport the sample to your lab in Lagos. So you need to put them in dry ice, not the normal ice cubes we do in our kitchen. So dry ice to make that your sample to actually remain the way it was from your site of collection. So carbon dioxide has been shown to retard fungal rotting of fruits caused by a large variety of fungi. So that has been applied to apples and pear. But the mechanism of action of this fungi is not known. It has also been shown for many years that ozone added to food storage environment has a preservative effect on certain foods, and that one is released in parts per million to the food, and this has been effective against most of the spoilage organisms. So it's like spraying ozone on the fruits, and that ozone will now serve as a preservative so that spoilage organism will not be able to enter into the food. So since it is a strong oxidizing agent, as ozone, it should not be used on high lipid content foods because it will cause an increase in rancidity. When you say something is rancid, like oil can be rancid in the mouth, that's biochemistry. There's a breakdown of some hydrocarbons, some chains, and it will not change the taste of that oil. Begin to, I don't know another word to express rancidity. Then your blue band or margarine can also be rancid. So because both carbon dioxide and ozone are effective in retarding the surface spoilage of beef under long-term storage. So all these things are actually applicable in developed countries. But in our country today, we are still struggling with the issue of herdsmen, not to talk of how to even preserve the carcasses of the beef. So whatever they sell to us is what we are eating. So there are examples of cardinal temperatures for these microbes as stated here: thermophiles, microphiles, saprophytic, facultative saprophiles. This should come under temperature, please, not under concentration of gases. So that takes us to, so the last group of factors that affect or influence microbial growth in food is called the biotic factors. And these influence microbial growth in food. And the first factor under that is growth. In a food environment, an organism grows in a characteristic manner and at a certain characteristic rate. So the length of the lag phase, the generation time is the doubling time, and the total cell yield are all determined by genetic factors. So accumulation of metabolic products may limit the growth of a particular species. If the limiting metabolic product can be used as a substrate by other species, these new other species may now take over, either partly or wholly, creating what is known as association or a succession. Now, what happens under growth is that, they exist as a community. And in that community, it is survival of the fittest. All of them are growing. All of them needs food. So the food that is available, for example, let's use the batch culture as an example. Within that batch culture, you have different types of microorganisms. You have Staphylococcus, you have Lactobacillus, you have Streptococcus in the same environment, and you supply food to them. Now, some just like some children, if you have 3 children, one out of that your 3 children may be a slow eater, and you now put food in a tray for them together to eat, and you have one that is a very fast eater. The very fast eater is the one that will get filled up on time, and the slow eater will only have a very little quantity of food to manage with. That's exactly what happens in that community of growth. Now, as they grow, they release metabolites, as explained in our microbial growth curve. Now, that metabolite that is being released, I said some of them are acids, some of them release acids, some of them release gases, some of them release antibiotics, some of them release just skipped my mind. About seven things. I'm going to get to that note anyway. Now, so in that same environment, that antibiotics will kill some group of microorganisms, while another group in that same environment will use that antibiotics as food. You understand? They will use it as food. So it is not a nutrient to them. So the gases that are released, to some it will choke to some of them and die. Some other one will use that gas as a cofactor to actually synthesize another thing for themselves. So at the end of the day, a particular species of microorganism will be the one that will dominate, and that's what we call succession. So if you put those children in one room and you keep giving them one bowl of garri or one bowl of rice every day, and the fastest eater is the one that eats up the food, and over time the other one that gets just little to eat will grow thin, be emaciated, and God forbid, if care is not taken, that person can even die. Of course, the immune system will not be okay, not be sound, and that person will be prone to all kind of infection. Now, so due to the complex of continuing interactions between environmental factors and microorganisms, a food at any point in time has a characteristics flora, and that is what is known as its association. That's what is known as its association. So the microbial profile, which changes continuously, resulting in one association succeeds another, is what is called succession. I've explained that. Now, in the microbial deterioration and spoilage of foods, this phenomenon has been observed repeatedly. So as long as metabolically active microorganisms remain, they continue to interact so that dominance in the flora occurs as a dynamic process. So based on their growth enhancing or growth inhibiting nature, these interactions are either antagonistic or synergistic. It is the growth of this microorganism in a particular community or environment that will actually lead to antagonism or synergistic. From the word antagonist, they are enemies. Synergistics, they are friends. Now, antagonism. Another biotic factor that influences the growth of microorganisms in food is the competition for nutrients and other factors which promote growth. In the same way that humans compete for food when kept together within the same environment, microorganisms also compete. So in food systems, antagonistic processes usually include competition for nutrients, competition for attachment, that's the adhesion sites, unfavorable alteration in the environment, and a combination of these factors. So an example of this antagonistic factor is raw ground beef. Even though Staphylococcus aureus is often found in low numbers in minced meat, staphylococcal enterotoxins is not usually produced. So the reason is that Pseudomonas, Actinobacter, and Moraxella association that is always present in ground beef grows at a higher rate, outgrowing the staphylococci. Can you see that? So because the other three genus that are present also in ground meat, they grow at a very higher rate than the staphylococci, so they will overshadow it. So the staphylococcus does not even have enough nutrients to eat, not to talk of now releasing enterotoxin that will kill the other ones. So is it not what you eat that you will use to fly? So organisms of high metabolic activity may consume required nutrients, selectively reducing these substances and inhibiting the growth of other organisms. So depletion of oxygen or accumulation of carbon dioxide favors facultative obligate anaerobes, which occur in vacuum-packed fresh meats held under refrigeration. So staphylococci are particularly sensitive to nutrient depletion. Coliforms and Pseudomonas species may utilize amino acids necessary for staphylococcal growth and make them unavailable. So other genera of Micrococcaceae can utilize nutrients more rapidly than staphylococci. So streptococci inhibit staphylococci by exhausting the supply of nicotinamide or niacin and biotin. So staphylococci is a poor competitor in both fresh and frozen foods. So at temperatures that favor staphylococcal growth. The normal food flora offers protection against staphylococcal growth through antagonism, competition for nutrients, and modification of the environment to conditions which are less favorable to staph. So competition between these organisms may be affected by changes in the composition of the food and changes in extrinsic or intrinsic factors. So that is what we mean by antagonism. They produce something that will not allow other organisms in that same environment to thrive. You are oppressing them, just like the Boko Fulani herdsmen are now oppressing the farmers in Nigeria. It's antagonistic. So another factor is called metabolism. Metabolic products from one organism can be absorbed and utilized by other organisms. Changes in pH may promote the growth of certain microorganisms while discouraging others. A very common example is the natural wild fermentations used in most West African countries to preserve foods, in which acid production during the process establishes the dominance of acid-tolerant organisms, such as the lactic acid bacteria. So changes in water activity in the food can influence symbiosis. That's a win-win thing. At warm temperatures, Clostridium perfringens, if present, can lower the redox potential in the tissue of freshly slaughtered animals so that even more obligate anaerobic organisms can grow. Clostridium perfringens are mostly found in soil. So when you find them in slaughter animal, look around the slaughter area. So soil has contaminated the meat. There are some associations where maximum growth and normal metabolic activity are not developed unless both organisms are present. So these associations are known as symbiotic associations. For a man and a woman to produce a child, there must be a wee thing. A man alone cannot, we have not gone to that era of cloning. So a man will supply sperm or semen, the woman will supply an egg, and fertilization will take place. So it is you drop, I drop, 50-50, symbiotic is an agreement. So the metabolites that a particular group of organisms is synthesizing favors another group of microorganisms. So they work hand in hand. So in conclusion, the intrinsic factors of the food which affect microbial growth rates factors are related to the food itself. And these are pH, nutrients, water activity, redox potential, the presence of antimicrobial agents, temperature is also there. While the extrinsic factor are related to the environment in which the food is stored. And these are temperature of storage, composition of gases, and the relative humidity of the storage area. So this brings us to the end of the factors that affect the growth of microorganisms in food and the end of our module 2.